I'm Ushma Neal with the Journal of Clinical Investigation, and I thank you for joining me for the next in our series of conversations with giants in medicine. I have the pleasure of speaking with Bonnie Basler from Princeton University today. Basler has revolutionized the way that we think about microbiology. She elucidated the chemical language that bacteria use in a process called quorum sensing, which allows bacteria to count their numbers, determine a critical mass, and change their behavior in unison, resulting in virulence or sometimes bioluminescent. She's been called everything from a scientific dynamo to the bacterial whisperer to a MacArthur genius. As well, she excels in the art of scientific communication, has dabbled in theater, and most mornings you can find her teaching an aerobics class in Princeton University. Thank you so much for joining us today, and I really look forward to hearing a little bit about your very unusual path in science.
Delighted.
Do you think you could start by telling me a little bit about your upbringing and what you were like as a child?
Sure. I have a regular childhood, sort of middle-class family, no scientists. I'm the middle kid with an older sister and a younger brother, growing up in the 60s and 70s. And I think you're asking me, how'd you end up a scientist, right? And I loved nature, I loved animals, I loved being outside, I loved doing puzzles, I loved figuring things out. And I think I had these sort of enlightened parents that probably like all parents were trying to give me opportunities. So I took piano lessons, and that was a disaster. But I did work as the assistant to the veterinarian at the zoo, and that was, I came home overjoyed every day. And so by sort of following those kinds of paths, I sort of ended up in biology, loving biology. And then I went to college. I went to UC Davis, which is a really famous vet school, with the idea that I was going to be a veterinarian, because in my generation, if girls, showed an inkling for science, there were, you became a nurse, you became a veterinarian, those were very acceptable sort of routes for girls to go into. So I went to college to be a vet, and that lasted about 2 weeks because when I got into the classes, I realized instantly that what I really love are live animals. And so I got to college and I was pretty lost with what I was going to do, but what was wonderful for me was that as part of this sort of track to be a vet, you started taking biology and biochemistry, and they didn't call it back then, but it was molecular biology, and I just loved those classes, so I stayed in that track.
You did some work during college on E. coli neuraminidase.
Yes, I did. This is ancient history now, but yeah. So again, going back to this idea that I had gone, since I had been 10 or 11 or 12, all I had ever thought about was being a veterinarian. And then you get to school, and within, a month of arriving, there was no plan B. I had no plan B. And so I was very lost, but I was, as I just told you, enjoying these classes. And so back then, this is all before the internet and stuff like that, the professors, there was a little, piece of paper with different possibilities in labs. And so this professor named Dr. Troy in the medical school had these two projects. One was on cancer, it was actually on Epstein-Barr virus and cancer, and then one was on E. coli. And at this point I was a junior in college, and my mother had died the summer before, and I was committed to curing cancer, for very personal reasons. And so I saw this little blurb, and I went to him and said, I want to volunteer in your lab, and I'd never even held a pipette or anything. And so he took me in, but he put me on this E. coli project. And I was like, wait a minute, I have zero interest in that project. That can't possibly be interesting. Cancer's the interesting project. But I didn't have any skills. And I was a volunteer, and he'd already said yes. And so I thought to myself, I was very earnest, and I would try really hard, and I would just prove to him that I was good enough to be put on the other project. And so, of course, I fell in love with these little critters and how you could find out so much about them and in such a short time. Like what I realized through my whole life is that microbiology just matches my personality, right? I'm very high energy. I'm very restless. I have zero patience for a lot of things. And so this idea that you could have a surprise in the incubator every single morning, in some way it's it's a sickness too, right? Because you can have a surprise in the incubator every 8 hours if you planned it right. But for me, just the sort of how rapid it was to do an experiment, that fit for me. And then the other thing was later, the sort of simplicity of the systems were something that my brain could manage. And I loved the morass of that confusion where you don't understand what the results are for a long time and they're kind of rattling around. I still love that. And then I just never looked back. And I've never worked on anything except bacteria since I was 19 years old.
Right, so you moved on to Hopkins for
Yep.
your graduate work.
Yeah, so Dr. Troy had, see, I still call him that because I was a 19-year-old. So he had done a sabbatical at Johns Hopkins for the person, Saul Roseman, who would become my graduate advisor. And so this is all long before, you flew out and interviewed. You just got a bunch of, just some acceptances and rejections. And so I had this handful of yeses and nos, and I got this yes from Johns Hopkins, and I remember, this is a quote, Dr. Troy said, throw the other envelopes away. And I did, right? And so it was not a sophisticated decision. I think that's going to be a theme of this interview, that I do things impulsively, and I do things, back then I was not sophisticated about looking at what a program offers, but we didn't have all that information at our fingertips, right? And so I had this advisor, he's my advisor. I took his advice, right? And so I went there and worked for Saul Roseman. Again, this was a bacterial lab. And going back to what you asked me about, we had worked on neuraminidase in E. coli. So both Troy and Roseman worked on, how the sugars on the outer coat of the bacteria, how they got assembled, how they got put there, how they decorated the bacteria. And so Roseman was a bacterial sugar biochemist, and I went there to work on that project, which was how sugars, in this case, got metabolized and processed. And then it wasn't going anywhere, my project. I was about probably 3 years into my graduate work, and right around then, Roseman, I guess, had written a grant to the ONR, which is the Office of Naval Research, who funded back then a lot of basic research in the ocean. And so they were interested in how what we now call biofilms got on boats. So bacteria attach to the boats, then the barnacles and everything get on, and then it just drags the boat down. So it costs the country $1 bazillion in gas. They have to take the boats out and scrape them off. That's the Navy relevance. But the first step was that the bacteria attached to the surface. And so I remember one day, my project was going nowhere, and Roseman brought this little vial of this next bacterium that was called Vibrio farnesii. It was a little stab. And he brought it in and he handed it to me, and he goes, here, this is your thesis project. And so I streaked it out, right? And so I started to work on that. And what I found was that the bacteria chemotacted to a particular sugar, glutanac, which is the most abundant sugar, and that leads them to the surface, and then they attach and they start to metabolize the surface. Now we actually have names for all these things, but what I got fascinated by was how these bacteria were perceiving their environment, right? And then doing the right thing. In this case, they swam to the surface. They didn't swim past it. They swam there. They recognized the goodies are over there, and they would go. And I thought back then, that's so cool that bacteria, in some way have some kind of memory, right? They know it's better over here than back there, right? And they do something about it. And so this, for me, really was the beginning of and of course I'm maturing as a scientist during this time, but it was the beginning of my, starting to appreciate in some fundamental way, the bacteria have these amazing sensory perception systems, that they have fantastic behaviors, right? And I like the idea of these marine bacteria, right? They're really old. The ocean is a nutrient desert, so these bacteria I thought were smart, right? And so I really got into this idea that you could study how an organism perceives and responds to its environment by studying a bacterium.
And it was at an ONR conference where
and so then, as part of that, I was, now we fast forward, so I was almost done, so I was in my 5th year of graduate school, and the ONR put these little meetings on where they made all the PIs come and talk about, it was like progress reports. And so this was right at the time that they had just built this, it was called the Center for Marine Biotechnology, and it was on the pier in Baltimore, right? So I'm in Baltimore, a mile up the street at Johns Hopkins, and the meeting was going to be held at this new marine center a mile away. So I begged Rosamond, because I'd never been to a science meeting before. I'm like, look, there's no airplane. I just drive my car down and drive back, and I'm going to hear some science talks. I never even heard really. I'd gone to seminars at in the department and stuff. And so I thought, this is going to be great. It's going to be all this marine stuff. And so I drove myself down in my Ford Escort to this meeting and listened to the talks. They were all these 20 or 30-minute talks. I can't remember that. But then this story of the next step in my career. So Mike Silverman was one of these PIs that had Navy money, and he was working on bioluminescence. And I think from the Navy's point of view, the Navy so bioluminescence is all over the ocean. Almost everything either makes or uses somebody else's light in the ocean. The Navy cared about that, right? Because, if your submarines are going or the Navy SEALs are jumping in the water and, there's all this bright light, they cared about it both to use it and then to be able to not have it there if they didn't want. That was the relevance, and I think why they were interested in this bacterium. And so Silverman was a geneticist. So I should say a few things. So my undergraduate and graduate work, I was trained as a hardcore, grinded up sugar, carbohydrate biochemist. Make fine proteins, purified proteins, sugars. I looked at the degradation products of this glutamac polymer, right? It was hardcore biochemistry. So I was already thinking about getting my postdoc, and I felt, this is 1990, and I felt very crippled because I had never used a restriction enzyme. I had never cloned, this is not 1970, this is 1990, right? And so I confused genetics with molecular biology, but I didn't know either of them, right? I didn't know what a gene was, a promoter, a transcript, none, zero, right? So that was already in my mind. So I go down to this meeting, this conference, and Silverman gives this talk about this bacterium Vibrio fischeri that he had been working on at that point. And he, with a graduate student in his lab, Joanne Ingebrecht, had shown, I should go back. Woody Hastings, 40 years ago, had shown that this bacterium, when you had it and it was dilute, when it was at low, it didn't make light, but when it grew to a certain cell number, all the bacteria turned on light together. And they had already shown, this was in the 70s, that if you took the cell-free soups, you could squirt them from the dense cells and put them on the dilute cells. The bacteria would make light, and they had purified a molecule. And that had been known since the 70s, not known by me. It was sort of this amazing finding that Hastings had found that was just sort of lost. I don't mean to say anything bad about Woody, right? It was a seminal piece of work. But I think because it was on this goofy bacterium, that glowed, that wasn't a pathogen, that nobody had ever heard of, it was just kind of a finding that was out there. But Silverman knew about it, right? And so Silverman's a geneticist. And so, and this, you also have to remember, is all before GFP, it's all before genomes, it's all before microarrays. We were doing, old-fashioned genetics back then. And so Silverman realized that he could exploit this bioluminescence to find the genes that gave the circuit, that told that bacterium when to make that molecule, how to detect it. He'd figured that out. So anyway, that's in his mind. But so me, I'm up to 25 years old, or however old I was, and Silverman gets up there and tells the story from Hastings, right, that this bacterium.
And it was an awakening.
It was my only epiphany of my life. But so Silverman gets up, this little guy gets up, he gives this talk, he tells this story that the bacteria don't glow like this, then they do glow when they're in a group, and then he knew about the molecule. And anyway, so he gives this little talk, and I'm just sitting there, and I had never heard anything like that in my life. We all, meaning everybody and the microbiologists, all thought that bacteria were these asocial recluses. They just divided in half, and each cell did its own thing. And here's this guy saying, no, the way they do it is by doing this bioluminescence together. And I just remember thinking, that is either the coolest thing I've ever heard, or this guy's a nutcase. So Silverman gives this talk, and I just thought, that's amazing. The bacteria are acting as a group. And remember, this fits into Bonnie's idea of how do bacteria sense the world, right? I'm working on a similar, a different behavior. And then I also thought at that moment, and then the rest of his talk was all about the genetics, finding the genes, and I didn't understand any of that. But I did get that the way he did the screens was you made mutants, whatever that meant back then. You turned the lights off and you looked for bacteria that were glowing when they shouldn't be or not glowing when they should. And I thought, I am smart enough to turn the lights on and off in the room, right? And figure out which ones are dark, right? So I literally, when his talk ended, I ran up to the podium and I said, you just have to let me be your postdoc or I'll kill myself. Something like, not the best, interview strategies, but I literally and said, you just have to let me come work for you. And so eventually he did. He's a very shy person and I'm not, right? And so I think he probably worried that this might not have been the best match, right? And then the other thing is that he took each of us in his career, he took each of us one at a time, right? And so we would come, so he only had, 5 or 8 postdocs in his whole career, right? And he would take each one of us and we would work on something big, he and you, right? And then you took it all and he would start on something else, right? That is the way science is not done anymore, right? We all have more, it's a bigger machine now. We all have more people so that when a postdoc takes something, I still have something, but he just, I remember so then I got to his lab and I remember the first day, he just said, what you get is what you take. And I'm like, where are the pipettes, right? And so he was the most amazing mentor because he was so generous. And also, right, most of the time I was the only one in the lab. So it was there was a technician, Miriam, right, who'd been with him for 20 years. And so the 3 of us literally worked together for 4 years. And it was just this utopian postdoc because I got there. Remember, I don't know what a gene is. I don't know what a restriction enzyme is. He's like, let's do a transposon mutagenesis. And I'm like, transposon? Like lambda. I never heard of any of that, right? And then it turned out that I worked for 4 years with one of the world's greatest living geneticists.
And it was during this time you both came upon the second auto-inducer.
Yeah, so then what I went to, so Silverman with this graduate student, Joanne, had kind of worked out the beginnings of this Vibrio fischeri circuit. And so when I went there, the project we decided to do was to work on this cousin, Vibrio harveyi. And the idea was, so Vibrio fischeri is this symbiont. It lives in a squid. It lives in this very pristine environment. It's the only bacterium in there, right? It's coddled, right? And so Silverman and Joanne had figured out the enzyme that made the molecule, the receptor, and how this whole circuit that they call autoinduction worked. That's what the talk I heard was. So they already did that. And so what we decided to do is to work on this free-living bacterium, Vibrio harveyi. It was clear it was going to have quorum sensing. It only turned on light at high cell density, but it made light in the free-living state, and it lived free-living in the ocean. So our idea initially, which isn't what was that there would be more sensory inputs controlling bioluminescence than just this molecule, right? And again, remember, that's what I want to do. How do you respond to a changing world? So I was going to come in and just quick, do a transposon mutagenesis and knock out the genes they had found in Vibrio fischeri, which were called LuxI and LuxR. So I was going to just look for some dark mutants that didn't have that, and then we wouldn't work on that, and we'd figure out how other inputs fed in, like oxygen, like iron, like different things that we knew regulated bioluminescence. And I get there and I start doing these transposon mutageneses, and I made a gazillion mutants, and whenever they were dark, they had a mutation in luciferase, right? The enzymes that make light. Like, I could not find these genes. And of course, so you have to put yourself in my place, right? I'm like, okay, I'm just not getting genetics, right? He keeps telling me to make these mutants. I do all these controls, but I can never find these genes. And so that took a long time because my expectation was to find what was in the related bacterium. And then finally, at some point, and I have no clue how, eventually, you sit down and you think like, okay, stop doing the same thing over and over again. What is the interpretation here? And so the interpretations that I could come up with at that time was either the genes for making this molecule, make them turn on light at high number were essential. You're not going to get them if you make a transfer, or they were redundant. Like, okay, maybe I had two epiphanies where I was like, if there's another set of these genes, you knock out one, the others still work, they're going to be bright as wild type. So then I started to do more sophisticated experiments to try to look for bacteria that were dim, right? That just didn't, right? And by doing experiments like that, I never found the genes that they had found in Vibrio fischeri. Vibrio harvii doesn't have them. But what I did find were two circuits, right? So that worked in parallel. So there was a molecule and a receptor, and if you knocked it out, there was another molecule and another receptor. And so what I found was this completely different circuit that was much more sophisticated. So I never got, and still haven't gotten to how all these other sensory inputs feed into Vibrio harvii, because it turns out you could just do it all in what we now call quorum sensing, because there's all these molecules that the bacteria are sensing and responding to that say something about your number and how, well, we can talk about later, how related you are to one another, right? And then at the moment that I was finding that and publishing it, in other species, people found what Silverman had already found. So there was this year in 1993 and 1994 where all my papers came out saying, all my papers with Mike, right, saying there's two systems. And then there was a set of papers in terrestrial bacteria, pathogens, that said they have this Vibrio fischeri circuit, right? So there was this amazing
groundswell of understanding.
Totally. And so on the one hand, this Vibrio fischeri, this anomaly that had been around for a long time, because Woody found the molecule in the 70s, Joanne and Mike cloned the circuit in the 80s. It's 1993 or 94, right? And then there were 3 papers that came out that all found that circuit. So that was great. But then, by contrast, I didn't find that. I found that actually it was going to be even more sophisticated than that because there were 2 molecules. There's more now, but right. So then those papers helped me so much because it brought it out of the world of just bioluminescent bacteria. It validated working on the bioluminescent bacteria, and that my luck that I didn't find what all of them found, but found this much more fantastic, if you will, circuit in Vibrio fischeri. It just made the moment to me, then there's a field. Right?
So then riding on this wave.
This little tide pool. Yes, this wave.
Riding on your tide pool.
Yeah.
I read that you applied for
20
a
zillion
jobs,
or 20 or 50, 30, 50, yeah.
Got two interviews and then landed at Princeton.
Yes, so, right, so then I think about that a lot, right? So you know how this goes. You write the same application and then you change right? So what did they see that the others didn't see? Right, and then Princeton, remember, this is a bioluminescent bacterium. It's never even heard of fish, right? So Princeton does very basic science, right? We don't have a medical school. We're one of the few schools in the country that don't have that, and everybody in my department works on how sensory information from the outside gets in, whether you work on a neuron, whether you work on an embryo in development, whether you work on a cancer cell that's all goofed up, right? This project really does fit in the heart.
But you've stayed for 20 years. What has it been about that environment that's kept you there?
Well, so there's a debt, right, that I'm not clear I'll ever repay. That's why, so why are you interviewing me, Ishmael? It's gone great. Okay, so you don't break something, it doesn't need fixing, right? So there's 2 things, there's a lot of things, right? So one really is incredible devotion, right? Because there's more to it than just, so they hired me, right? And then we went on to find all these parts and work on quorum sensing and all different bacteria and find a lot of molecules and stuff like that, right? And the field has exploded. There's hundreds and hundreds of quorum sensing labs now, right? So it's been a great wave, right? But then to really answer the question you're asking me, so when I got there, right, there were these, remember, you want to say it's a way, Princeton, Harvard, there were these few papers, 4, specifically 3, and then mine. Well, I had a couple papers, but they were all on my circuit, right? And I couldn't get a grant. That's not really true. The NSF, I got a Young Investigator grant right off the bat. The NSF has funded me my entire career, and the ONR gave me some money, right? A few years later, they came back in and helped me with that. But it took me 13 years to get an NIH grant. And so you get tenure at 5, right? And so this business of the debt for me really is real, because a lot of places, I had a little Young Investigator grant. It didn't even come with overhead, right? So I was not helping the department. And then what Princeton would do is they would give me the graduate students and put them on a fellowship, or if ever a little cash came in for a postdoc, they'd funnel it my way. Tom Silhavi is my fantastic mentor. There was my next door neighbor, and I would just go down with a cart, and load it up. And I remember one night he caught me. It was midnight, and I was up on the top shelf, taking all the flasks and boxes of tubes. And he was standing outside the door. He goes, "Just don't fall off." Right? And so they underwrote me. When I came up for tenure, I had almost no money, right? Like not, they're used to people getting Pew and Burroughs. I didn't get any of those fellowships, and I just had this little NSF grant, a little Navy grant, and most people would be measuring my office, and right? And so then they made the decision that they would risk the fact that I might not ever be able, to get traction. And they still gave me tenure. And the decision they had, they said, this is a brand new way to think about bacteria, because I'm out there, come on, they work as collectives, this is how it's got to be. This is this young woman, back then, trying to overturn 400 years of the way people think about bacteria, right? And they sort of made the decision that we like the science, they'd read my papers, this is going to take time to percolate. And then they took me on for life. That's why I'm still there, right? Because I still feel I'm repaying that. But I don't want to act like I'm Mother Teresa here, right? The science has also gone great. And so then the other part of why it is a joy to be at Princeton is we're this tiny little school, right? No medical school, no other. Everyone works on basic fundamental problems, no matter whether you're in biology or physics or chemistry or engineering. Our engineers don't really make things, they make knowledge.
You talked a little bit about your students. So I read a profile about what life was like in your lab and some kind of off-the-wall characters. So how do you build your quorum, and what's your approach to mentoring with your students?
Right, so it's the Wild West. So so I always thought, I would figure that out, right? But the truth is every one of them comes with different warts and pimples, right? I have my own quirks. And each one is different. It's like a kind of big, slightly dysfunctional family. And I do think that all labs, at some level, take on the personality of the PI, right? So my lab is very loud, right? Lots of jokes, lots of laughing, lots of yelling, but all in jest, right? But then on the other hand, it's really positive about science, right? I think the thing that Silverman taught me, he always made me believe I was going to get there, right? He always was like, you're just around the corner, and oh, yum, Bonnie, I know you're going to figure it out. And I'd go back to the bench, I'm going to figure this out. He didn't have a clue, right? And I realized that motivated me a lot. So I am very positive, right? And then I think everyone is positive to one another. We all share everything. Ishma, it's quorum sensing, right? I think my lab already selects that people don't, we work on the most ancient collective behaviors and how the group gets more than the individual. So the lab already selects for a certain kind of person. If you're like real selfish and mean, you would never choose this as your project. It's about sharing public goods, right? So already I think just intrinsically, the kinds of people that come to my lab, like to do that. And so we all share everything. And I think also like in terms of your real question, right? I think when I was back, in the lab at the beginning, it's your job to set the bar, like how hard you're going to work. And it's not like I keep, how hard you're going to work, how much fun you're going to have, how respectfully you're going to treat one another, how positive you're going to be, and right. And now I don't actually. That's just understood, right? That we will all behave in that way. and they all do.
Now, you've mentioned before that you think the next generation antibiotic is maybe 5-10 years down the road.
Right. So I'll just tell you where we are right now, right? So we have molecules. So we screened for molecules that shut down quorum sensing. And we have molecules, if you are a worm, we can save you. If you are a cell in a tissue culture dish, we can save you with our molecules. We have we have some things that are starting to work in mice. And so those kinds of experiments, right? So these are anti-quorum sensing molecules, right? So they're antagonists of the real autoinducers, and
in models of cholera,
Pseudomonas aeruginosa, right? And so the Pseudomonas aeruginosa, we've done worms and we've done tissue culture cells, lung cells, because that's Pseudomonas infection lungs, among other things. We know we can shut down biofilm formation, right, with Pseudomonas and with cholera. And so those experiments give me confidence, to at least pursue this line of reasoning. Can you ever, every antibiotic we've ever had slows the growth of a bacterium or kills it. This doesn't do that. It would make them so they can't count, and then just give time for your immune system to mop them up, right? That's an untested idea. Right? And so I don't know if that can ever work. But we're sort of marching the path to get there.
Well, then what do you feel about the proliferation of Purell machines?
Yeah, so I have
everywhere?
a real problem with that. You'll probably get sued after this thing, right? So I have two problems with that. Number one is that it's soap. It's expensive soap, right? So I have a problem with marketing this as this soap is antibacterial, right? It pops bacterial membranes, right? So it's very clear you need to wash your hands when you're preparing food and after you go to the bathroom in this country, right? That is when you need to wash your hands, and soap is fine, right? So I have a problem with making something seem like more than this old-fashioned thing, because I feel that's marketers preying on our lack of knowledge, right? And then I don't care if people use it, right? I just think they're wasting their money, and I do worry about this. There's whole aisles of Purell now. I worry because what I wish people would realize in this country is that most of the bacteria that they encounter are keeping them alive. And I think that kind of a marketing strategy, sort of raising fears of germs, right, is really wrong, because what we really want people to do is to learn about all the great things bacteria do, because they are keeping us alive. And so I worry about that in terms of education a lot. And I also worry that it skews what really happens in the world. If you live in this country, a developed country, your chances of dying of a bacterial infection, are really, really low. And yet we're doing this Purell thing, versus if you're in an underdeveloped world, you will die of a microbial infection, right? And so I just find it amazing here that there's this incredible paranoia about germs, when that has nothing to do with our lives, because we have clean water, we have clean food, right? We have an infrastructure that keeps it so, versus the rest of the world that's really dying from these kinds of infections. I just find it amazing.
You've been called the bacteria whisperer by Neil deGrasse Tyson. I noted you got a standing ovation at your 2009 TED Talk. So you clearly, and even just through this interview, you clearly excel at making complicated scientific concepts easy to digest and understand.
I work on communication. It would be bad if I couldn't talk about it, right? Right?
Well, so tell me what you think. Would you ever consider a second career as a pundit?
Right. So I've actually, so this would be something I probably never said out loud before. I've actually thought about that. I've thought about a number of, these are called midlife crises, I guess, right? And I am old enough to have those. I've thought about a number of these second careers, right? Like what does it mean when it's the end of my life? Right? I have this legacy, I already know that, of these students and postdocs, right? I am so happy about that. It has been an honor and a privilege and a joy, right? And so that sort of happened, right? And so then you sort of ask yourself, are you going to find the next molecule, the next circuit, right? Or would that be enough, right? Or do you want to just do something else? That's what you're asking me. And so I've sort of dabbled in these things, right? And I do have this passion, like we're just talking about with this Pure. I have this passion for this country especially, which is that I worry so much, why do people hate science? Why do they think it's too hard to learn? Why do they think it's boring? And why do they think it's bad? And that's part of it. And then the other part is, you know, I do science because the taxpayers say I can, right? And so this idea that people, if people don't vote, and I mean for basic science, for letting a young woman back then working on a glow-in-the-dark bacteria that nobody, not she, not anyone else, could ever even imagine might make an antibiotic or a coating so you don't get a biofilm when you get an intubation device or a catheter or a paint or whatever is going to come from these sort of applied quorum sensing strategies, right? I did not have that in my mind then. I have some of it in my mind now, right? Like there has to be a place in this country to let that person, just driven by her curiosity, let it go where it's going to go, right? And so my worry is that the next young person doesn't get the shot that I got, you know, because this country doesn't understand that there's nothing to apply to make our lives better unless some scientist works on the basic part of it. And I mean that for everything, for engineering, for technology, for biology, for all of it. And so I've thought a lot about going into politics, right? Like could I actually work in sort of politics and education, right? To sort of change how people learn science and their appreciation of what it does and what it will do for the future of this country. And then I've also thought about this, you know, who are the scientific heroes, right? Like, so I think Neil deGrasse Tyson is this country's right now. You know, I think Carl Sagan was, you know, but who's the next one? And is it me? You know, I don't know, right? I just don't have any training for it. And so part of it is I don't know how to make that leap, because what I really know how to do is what I do. So I don't know how to make a leap into either of those professions, so I do them sort of as a volunteer, like today, like a one-off. And then the second thing, what I really will say is whenever I go do any of those things, you know, I enjoy it, but I find myself still running back to my lab, because the thing I love the best is when some young student says, look what I found, or even bad or good, I am the second person to know that finding. And so somehow, at least for now, that still gives me the same rush as when I was doing it with my hands, my own hands, right? And the same feeling of like why I went into this in the first place.
If not science and science communication, what else do you think you could have done in your life? Would you have gone on to be a veterinarian, you think, if you had the right teachers?
No, I think I really did have the right teachers at that moment, right? Because like learning what you don't like, I think is also as important as learning what you do like, right? So they helped me a lot. What would I be if I weren't a scientist? I'd like to be a movie star. I'm a ham, right? I really would. I like to, I don't think I'm gonna let you put this on this thing. No, I would, no, so I love, okay, so the real truth is, right, I like, you know, giving seminars, I like talking about things, I like people, you know, this sounds arrogant, I don't mean, I like people paying attention to me, you know, and like being engaged with me. And so I guess I would pick some other career like that, like probably a stand-up comic or, you know, something like that where you're in front of an audience. Like I get, you know, I do think when I'm, you know, giving seminars, you know, and it's performance art, right? It is, you know, there's a narrative, you know, and so I guess I would do something that actually that kept me in front of an audience. Right? But the truth is whenever I'm in front of an audience, if I don't have a picture of Vibrio harvey behind me, I'm frozen. And so this is big talk because I am terrified of all things, of talking about anything except science.
All right, well, thank you so much for joining me.
Thank you for having me. It was a lot of fun.
Good, great.